I will actually shift gear to vegetal cells and try to show you the virus capability that can bring the s-SNOM into the plant cell walls to study their physical and chemical property at the nanoscale. This is a collaborative work between two labs in Marseille, the Oak Ridge National Laboratory in Tennessee, and of course with NIASPEC. So just the context of the study is to try to develop some biofuel. So when you start from a lignocellulosic biomass of different kind, you can extract some byproduct. I listed some of it here, through some treatment. And in that case, we are interested in extracting cellulose from wood to actually extract the bioethanol or lubricant. So, of course, why we go to biofuel is because of the growing demand on fuel, especially for transportation. And we also have increasing interest in reducing the impact on the greenhouse of the burning fossil fuel. So one has to develop different alternatives of fuel from this lignocellulosic biomass. So as an example to illustrate this type of treatment, we investigated a chemical delignification process that we applied over a Poplus deltoid. We choose this type of tree because it has a fast growth, and so it's a good candidate for biomass investigation. It's also well studied, so we have lots of data access to make strong comparison when we go towards a nanoscale. So I introduce here the process. It's a three-step process. So you have the raw sample, you rinse it to eliminate the fat or the organics. Then we do an acetylation to remove some lignin, and finally we do an hydrolysis under acid environment to remove lignin and hemicellulose. So basically, we increase the solubility through this process of all species, the three main components, that is lignin, cellulose, and hemicellulose, through an acetylation process. Before going to optics, we already did some nano-indentation by atomic force microscopy. So I showed you here example of mappings of the elastic modulus, and we actually measured a reduction of this elastic modulus by a factor of 10 after the complete process, which is actually expected as we remove lignin and hemicellulose that actually are the link between the fibers, the microfibril of cellulose, and the lignin is the cement, it's like the cement of the vegetal cells. Now, the question is, can we correlate this nanomechanical data to chemical distribution and optical property at the nanoscale? Well, if you go to the literature, because of course you think of near-field optical microscopy when you want to do this type of study, we found out some work on aperture s-SNOM, where they, in the work of Keplinger, they could actually distinguish some shape of the cells. But if you look at the AFM images, you don't see much. You also have this nice work from the group of Pereira in IR-AFM, where they compare with TEM images at two different wavelengths here, and they acquired some spectrum in different regions of the cells. And we went to the s-SNOM to try to correlate the structure, the mechanics, and also the optical property and chemical property. So I will try to show you some correlation and also show you that we can actually monitor the delignification process. There is also, following our work, a nice work from the Ragoskas group on Poplus trichocarpa. I will not say too much because I think there is a talk after me in this session. So we start by doing imaging with the s-SNOM, and we actually did some colocalization of the different constituents. So you have here the topography, and actually we don't see much. Here you have the mechanical phase, and for now we don't know exactly what That means, and we acquired the optical images in reflectivity and absorption at cellulose-specific wavelengths and at lignin-specific wavelengths. And you can already see in the absorption that we can evidence some cellulose-rich region and lignin-rich, in that case here, region. So now, if we push the observation further, you can check that on the mechanical phase, we can have some correlation between the chemical distribution here and the viscoelastic property that is linked to this measurement, as we see higher value of the phase lag here that actually show that in the lignin-rich, we have higher elasticity. So to actually control this, we did some correlation between the mechanical phase obtained by AFM, and we did nano-indentation at the same time. And you see that when you have an increase of the phase lag here in the middle lamella where you have more lignin in our cell, you also have an increase of the viscoplastic index, that a decrease, sorry, of the viscoplastic index. That means you go towards more elasticity in this region. So that actually is a very good agreement with what we obtained with the s-SNOM. So this is the first correlation. And then we shift to spectroscopic measurement, of course, to try to monitor our delignification and try to see the lignin recalcitrance. So you have here the reflectivity images of the 4 samples following our process, and the spectrum associated along the lines that are marked in the reflectivity. So you have the cellulose peaks here, the lignin peaks, and the hemicellulose, and this is the region for the fat that is actually removed here. And the most important is going from this step to that step, where we actually see the band shift of the cellulose to higher wave number that is due to the acetylation of the alcohol link of lignin, cellulose, and hemicellulose. So we have all the shift of the band. We also have a new peak that appears here from the lignin ring. And finally, we also have all these peaks that are here from the ester COO link. And finally, the last step to try to remove lignin and hemicellulose, we actually see that the hemicellulose is removed, the lignin is removed, but we still have some remaining, let's say, peak associated to lignin. That means we have an incomplete extraction. So we have actually lignin recalcitrance through this process. And we know that since it's this peak, we have a good removal of the GS lignin, but we have low efficiencies of our process onto the G lignin that has a lower solubility. So actually the s-SNOM can give some information to improve this type of wood treatment. The last part of the talk is to go into hyperspectral imaging. So we stay in the fingerprint region of our polymers, and we pick the region from the mechanical phase where we assume to have a lignin-rich region, and then we acquired the reflectivity and absorption in hyperspectral images. And from that, we extracted the local optical property. So here is for a specific wavelength of the cellulose, and here is for a specific wavelength of lignin. And you see in the K mapping that we have a distribution from the cellulose and for the lignin that is different, completely different. And actually, the good thing was to see this mapping of K for the lignin that actually confirmed the mechanical images. So it's a good correlation between the mechanical and optical properties. Of course, since we acquired the hyperspectral, we also have the NNK of the cellulose-rich and lignin-rich region as a function of wave number. And the main thing was to try to see if we could have some information from this optical property for our delignification process. So if you go from the raw sample to elimination of the fat, you already see that the shaded region that is the standard deviation and reflect basically the variability, the environmental variability of the region, reduce when we remove the fat. And when we complete the process to the last step, you see that the standard deviation is quite reduced. And we also know that we come closer to the NNK of pure cellulose, since we are almost left over with cellulose. And it's actually a first measurement of this NNK in situ. Before that technique, we had to do extraction and then do a measurement of the NNK. And we actually know that we are not far from the actual NNK of the cellulose. As you see here, in the K, we represent the cellulose peaks, whereas in this region, all the lignin and hemicellulose aspect has been removed. So quickly to conclude, I hope I showed you a different capability of the s-SNOM to study plant cell walls and wood cell walls. I must say wood is a complex material that is anisotropic and has a hierarchic structure, which is also quite rough, so it's difficult to investigate these materials with near-field microscopy. Usually what people do is they embedded samples in resin and do ultramicrotomy to reduce the surface effect. But if you want to actually follow a wood treatment, you cannot really embed it into resin because you would modify the chemistry and also the mechanical property. So we also, of course, follow the delignification process, and we actually showed that we had recalcitrance of the G-lignin. And the main thing that we wanted to demonstrate is that we can actually have correlative information between the mechanical, optical, and chemical property of this cell wall at the nanoscale. Of course, we want to continue, and especially into treatment that use physical aspect instead of chemistry. For example, applying a tension to wood that would generate more production of cellulose. I want to acknowledge all my colleagues from the different institutes. I want to thank Philippe for his help and a constant replying to all my questions, the fundings, and you for your attention.